Based on what we remember from yesterday, what is it that is going to help cause an action potential in that skeletal muscle fiber?
A neurotransmitter?
We are going to have the connection between a nerve and that muscle fiber. And do you remember we talked about how the action potential would travel? Because there's only going to be one point on that entire muscle fiber. And remember, if we take a look at, for example, the sartorius, okay, that's the longest muscle of the body. And that means, depending on how tall you are, okay, for this sartorius muscle, that fiber could be this long. And somewhere on that fiber would be a terminal end from the nerve. So the point where the nerve is going to meet that muscle fiber is termed the neuromuscular junction, the NMJ. The neuromuscular junction, this is the view, or this is what it's going to look like. We say that this is a synapse. In between where we have that nerve meet the muscle fiber, we have this little area, the synapse, and there's a little bit of distance between the two. And this is going to be our synaptic cleft. Now, a couple of things that I want you to note about this coming together of the axon and the muscle fiber. What do you notice about the plasma membrane on that muscle fiber? Does it look just nice and straight?
No, it's depressed.
We're looking at it doing a bunch of hills and valleys. Located in, well, first of all, what would be the benefit of having the plasma membrane do that?
More area.
We have more surface area. What type of protein, what type of gate do you think is going to be located in this region? Ligand gated. In this region only. That's it. That's the only place you're going to find ligand gated channels. Why?
I guess because that's where neurotransmitters.
Do it, Brandon?
Isn't that because the neurotransmitters are right there?
All right, because when we look at this setup that we have right here, in this setup, we're going to say that there is a presynaptic terminal and there is a postsynaptic membrane. Well, the presynaptic is telling us the end of that muscle, of that nerve fiber. Okay? Because that's before the synapse. After the synapse is the muscle's membrane. All right? So we're going to have presynaptic, we're going to have the synaptic little piece, the cleft, and then the postsynaptic membrane. This is referred to as also being the motor end plate. Why do you think it's called the motor end plate? Think really hard. It's found on a skeletal muscle, because that's what we're talking about right now. Skeletal muscle gives us what?
Movement. Motors.
Movement. And Brandon, what'd you say?
Motor.
Motor. movement. So therefore, in the skeletal muscle, we're going to term it, also known as the motor end plate. Because it looks like a plate holding the food, right? So, at this point, okay, muscle cell fiber, it's innervated. The innervation from that nerve is going to be meeting at the area of the synaptic cleft. We will have presynaptic, we'll have postsynaptic. In the postsynaptic membrane, we've already determined that this is going to be full of ligand-gated channels. Up here, take note of what we've got. This is the end of that neuron, okay? How far away, just out of curiosity, is that neuron going to be? The cell body, the where we've got the nucleus and the ability to make those vesicles. Just out of curiosity. See if it blows y'all's mind like it blows mine. It's going to be in the spinal cord. So if you think about the end of that nerve, if I'm going to refer to my sartorius muscle, okay? My sartorius muscle, all right? The nerve cell body is up here. That means the axon has to make it here. Does that blow anybody else's mind? And remember, this is a cell. The only product a cell can make is a protein. The product of the nerve cell is a neurotransmitter that it packages in the little vesicles, and that vesicle had to travel that distance. Is everybody following me? Okay, so when we talk about the neurotransmitter for skeletal muscle, the neurotransmitter is acetylcholine, ACH. Now, the neurotransmitter is simply the chemical substance that the neuron made. It has to, well, it will travel down the axon to those terminal ends, and from that point, when we get an action potential, those little vesicles will begin to release their product. Their product for right now, for skeletal muscle, is acetylcholine. So, what does that tell me? Any ideas? This is my, if this is my nerve, am I on there? Okay. If this is my nerve meeting the muscle fiber, the way that they can communicate is this chemical. So, at the point where we're going to have the cleft, the post, okay, acetylcholine gets released. That tells me that the receptors, those ligand-gated channels on that cell membrane, are going to hold or be able to accept acetylcholine. Now, if that's the case, do it a different way. All right? Is everybody with me? Then the acetylcholine gets released. All right? And it fits into the ligands. The gate opens. And then my positives, my sodiums, are going to get to do this number. That means at that area of the neuromuscular junction, it will allow an influx of sodium. Because if the only thing that's going to be found in that region is ligand-gated channels, lots of sodium gets to rush in. Now when that sodium rushes in, what does it do to the charge? We make it more positive. Now, if we make it more positive and we begin to have that action potential travel the length of that muscle cell, that muscle fiber, that means voltage gateds are going to open, right? Why would we not want acetylcholine, which is the neurotransmitter here, to remain in that cleft? Why do we want to make sure that we get rid of it very quickly?
It might cause involuntary contractions or something.
If the acetylcholine remains in the cleft, what's going to happen to the gate on this? It'll stay open. Sodiums will continue to rush in. We'll continue to have voltage get affected. And the contraction, not involuntary, it could be, okay? But if that's the case, that muscle will remain contracted. Any idea what a term for that is? Tetanus. All right? Skeletal muscles can handle that for a small amount of time. Okay? Not very long. So what we're going to find in this cleft, you know how we always can break stuff down and it ends with ASE? That tells us it's an enzyme that will break that product down. We have acetylcholinesterase. Say that a couple of times. Acetylcholinesterase. And that can degrade the acetylcholine, and it helps prevent any type of accumulation of the ACH. We need that. Okay? So in the process that will take place, something will start an action potential. Something. Okay? I need a pointer. I need something to point with. I'll get one of my favorite muscles.